Hello everyone, welcome to my YouTube channel Microbiology Simplified. I am Dr. Amit Kumar Singh, MD Microbiology working as professor. This video is a quick recap video of the previous 3 videos which I made on the morphological forms of various protozoa and parasite. So let's do a quick recap. So let's see. So this amoeba, including intestinal and free living, this lumen dwelling flagellate and this ciliate, all these three have what is common in them is the cystic stage. Okay, so what is the difference between them? So cyst of B. coli is binucleate. Okay, cyst of Entamoeba histolytica is quadrinucleate, and the cyst of Giardia duodenalis is also quadrinucleate, that has got 4 nucleus. Rest, this free living amoeba, Entamoeba has got single nucleus, that these are uninucleate, and these two, that is the Trichomonas species, which is a genital flagellate, and Dientamoeba fragilis, which is an amoeboflagellate, these have got no cyst. Okay, no cystic stage. Now the second morphological form which is seen among them is trophozoite. Trophozoite is present in again both the amoebas, the lumen dwelling flagellates, the ciliate, and along with that it is also present in sporozoan, which includes hematozoa and coccidia. Okay. So what is the difference between these trophozoites? So again the trophozoites are uninucleate except in case of Giardia, in which it is binucleate, and in case of Vanadidium coli again it is binucleate. So both the cyst and trophozoite is binucleate in case of Vanadidium coli, whereas in case of Giardia duodenalis it is binucleate. Okay, so this is about the morphological form seen in the amoebas and the lumen dwelling flagellates. Now the morphological form seen in the hemoflagellate. In hemoflagellate, if you remember, the name of the morphological form is mastigote. So there are 4 types of mastigote. First is amastigote, second is promastigote, third is epimastigote, and fourth is trypomastigote. This amastigote, A means no external flagella, but they have got internal flagella called as exoneme, and these flagella originates from a structure called as kinetoplast. So on the basis of the position of the kinetoplast, we identify the rest 3 stages, that is promastigote, epimastigote, and trypomastigote. In promastigote, the kinetoplast is anterior to nucleus. In epimastigote, kinetoplast is again anterior to nucleus, but then the kinetoplast is slightly posterior compared to promastigote, and also it has got a short undulating membrane through which the flagella runs before coming out from the anterior end. Whereas in case of trypomastigote, again the kinetoplast is posterior to the nucleus, and this time nucleus is situated centrally, and it has got a long undulating membrane from which the flagella comes out, runs before coming out from the anterior end. Okay, so these are the morphological forms seen among the hemoflagellates. Now coming to the sporozoa. In a sporozoa, if you remember, there are 2 types, hematozoa and coccidia. Hematozoa includes the malaria parasite, that is Plasmodium and Babesia, and coccidia includes the opportunistic sporozoan parasites, which include the opportunistic parasite causing diarrhea and opportunistic parasites causing toxoplasmosis, that is toxoplasma. So these 3 causes diarrhea and this causes toxoplasmosis. Okay, but all of them are opportunistic protozoan parasite, which causes disease in HIV and immunocompromised individuals. Okay, so the morphological seen among them are somewhat common, overlapping. Okay, so just see what are common in them. First, merozoite is present in this hematozoa, merozoite is present in coccidia. Trophozoite is present in hematozoa, trophozoite is present in coccidia. Scisont is present in hematozoa but not in coccidia. So scisont is only present in hematozoa. Macrogametocytes, microgametocytes, these are also not present in coccidia. However, the microgamete and macrogamete are present in coccidia. And ookinete is again not present in coccidia, but the oocyst containing sporozoite is present in coccidia. And one more morphological form which is present only in coccidia is the tissocyst, which is not present in hematozoa. Okay, so why I made these two separate box for the morphological form of hematozoa? Because these morphological forms which is mentioned in this box is present inside human host, and these morphological forms are present inside insect host, and all these morphological forms are present inside human host in case of the first three, that is cyclospora, cryptosporidium and cystoisospora. Whereas in case of toxoplasma and sarcocystis, these are present in the, some of them are present in the definitive host in which sexual reproduction occur, are these two. First this microgamete and macrogamete, and then this trophozoite. Okay, trophozoite, merozoite, microgamete and macrogamete. That means sexual reproduction occurs in the definitive host or the vector. Okay, so these two, macrogametocytes and microgametocytes, these are not seen in coccidia. They directly produce microgamete and macrogamete. Okay, and this oocyst, again this oocyst in this coccidian parasites differ from each other on the basis of the number of sporozoites contained in them. So if you remember, toxoplasma and cystoisospora, these two have got 8 sporozoites, whereas crypto and cyclospora, these have got 4 sporozoites. Okay, but again the difference between cryptosporidium and cyclospora is the number of sporocysts. In cyclospora the number of sporocysts is 2, whereas in cryptosporidium the number of sporocysts is 1. That means in this case one sporocyst contain 4 sporozoites, in this 2 sporocysts, that means one sporocyst contain 2 sporozoites, that is total of 4 sporozoites. However, in them there are 2 sporozoites, sorry, 2 sporocysts which contain 4 sporozoites each, so total 8 sporozoites seen in them. So this is a difference, and also they also differ in the size. The biggest oocyst seen is among cystoisospora, which is around 25 micron, whereas the smallest is seen in cryptosporidium around 5 micron, and also the oocyst of cystoisospora is elongated or ovoid in shape, whereas in case of crypto it is rounded, cyclo it is uh, circular or rounded, and toxoplasma again it is oval to rounded. So it is elongated only in case of cystoisospora. Now the tissue cyst, which contains basically the bradyzoids which is present inside the either the skeletal muscle or brain depending upon the species. So these bradyzoids, again the bradyzoids are trophozoite in case of toxoplasma, whereas it is merozoite in case of sarcocystis. Okay, so there are more detailed information about these morphological forms if you'll go through the lecture separately. This is just a quick recap, so just to remember that which morphological form is seen in which protozoan parasites. So I hope this, if you go through this video, at least you will be able to recall the name of the morphological forms present in various protozoan parasites, which will help you to identify the life cycle, or if in the questions if the diagram is present and it is mentioned that it is a merozoite, if it is a trophozoite or it is an oocyst, so you may be able to find out that okay, if it is oocyst, so it will be present only in case of the sporozoan parasite, because oocyst is not present in either amoeba or in flagellates. So if it is mentioned cyst, so you immediately it will click in your mind, okay, it is if it is cyst, it is present only in amoeba or lumen flagellates or ciliate. So this is a quick recap about the various morphological forms of the protozoan parasites. Hope this will help you in the examination. All the best. Take care. Bye bye.